starting. Good morning everybody. I really would like to thank all of you for coming to this European Xylella conference in 2017. It is really my profound pleasure to welcome you all to the University of the Balearic Islands in Palma de Mallorca to this conference. And as you probably know, because if you are here, it's because you are very much interested. The bacterium Xylella fastidiosa is one of the most challenging pathogens we are dealing now, at this moment. Its introduction in Italy, as you know, and France, and very recently in Spain, and particularly here in the Balearic Islands, it's one of the most, as I say, challenging situations we are dealing with, because it's a bacteria, as you know, that is very important for the different crops. So as you could imagine, as a researcher, it's really an honor for us to hold this scientific meeting here at the university. We have in front of us two and a half days, very intense. Thank you to the program from EFSA, from 8 of the morning to 8 in the afternoon. So we really have a very intense program. I really wish that your stay here is going to be very pleasant, and we are going to have the opportunity to discuss a lot of very interesting issues in regards to Xylella. Just to be brief, because we are running already a little bit out of time, I really would like to thank to EFSA, and particularly to Dr. Giuseppe Stancanelli from Alpha Unit in EFSA, for giving us the opportunity to organize this meeting here at the University of the Balearic Islands. Also, my gratitude to the organizers from EFSA, Vanessa Desi, and also Francesco Bracci from Pomiglio events, for the great job they made for the organization. Even if they were physically here, it is difficult to organize a meeting by email and phone calls and so on. And also my gratitude also goes for all the team in EFSA, particularly to Sara Tramontini, Evelina Sevienzek, sorry Evelina about your surname, it's always difficult to pronounce, Alice Del Bianco, Emanuela Tacci, and as well as to different members of different European projects like Maria Saponari and Donato Boscia for the support, scientific support for this meeting. And a special thanks go also for the university for letting us to organize this meeting in this venue, and particularly also to the Fundación Universidad Empresa, the two ladies working there, Marga Omar and Veronica, that they gave also really a great support for this meeting. I also want to thank the administrator of this building, Michele Orrac, that he really facilitated everything to give us the opportunity to be here today, and also to INAGEA, to Dr. Ippolito Medrano, that is also supporting the meeting. And of course, I give also the thanks to the Vice Chancellor of Innovation, this is Dr. Jordi Llabres, for being here today for this opening. As I say, we are now addressing the welcome of the meeting, so I pass the word to Dr. Giuseppe Stancanelli from EFSA.
Thank you very much, Miguel. So, welcome to everybody on behalf of EFSA, but also, in particular, on behalf of the whole organizing committee of this conference, which is not only organized by EFSA, but is organized by us together with the two Horizon 2020 project, POnTE and XF-ACTORS, with Euphresco and the University of Balearic Islands and INAGEA. So, I will move there then.
It's just, you need to switch on.
Okay. So, just a little bit of background of why we're here. So after the outbreak in Apulia, in his first opinion on the Xylella fastidiosa risk for Europe, the planetary panel of EFSA recommended research on Xylella fastidiosa. This topic was also was object of a resolution of the European Parliament. And there was a series of workshops organized, a workshop by DGRTD, on option for control of Xylella fastidiosa was in July 2015 in Milano at the Expo. And following that, EFSA has received a mandate to try to identify the knowledge gaps and the research priority for Xylella fastidiosa research in Europe, and also to produce a periodical report, a state-of-the-art report on the results of research. And that's where this conference came from. In the same time, in 2015, specific funding for research was allocated for Xylella fastidiosa research under the Horizon 2020 project, and that's why we have now 2 European projects on Xylella, POnTE and XF-ACTORS. So the previous workshop was done in November 2015 in Brussels. Many people from the audience were participating to this workshop. It was a workshop really to identify which were the key areas for research. And we had 150 participants. The event report is online. That workshop was a networking also opportunity for researchers to establish research groups and networks. So this conference is a cooperative conference. It's organized with different partners. And this is a 2017 issue. We will talk about this on Wednesday. We also would like to organize periodically these events, also with other partners. And the organizing committee has already been acknowledged by Miguel. And also we should thank the scientific committee that helped us in building up the program that you have seen today. So the program is very dense, therefore, welcome. We'll try to keep it very short. There will be a focus from the chair and the speaker to stay on the time, so to allow sufficient discussion at the end of each session, and to try not to shift to the time of other sessions during the program. Particularly today is very dense. Tomorrow also, for the whole day, till the poster session, until 8:30. And we'll finish at 1 o'clock on Wednesday. You all have the program on the website or in the app. So some short rules. Please switch your mobile phone if you didn't do it yet. I did my one right now. You have a Wi-Fi connection available in the room. And also, in case of emergency drill, the colleague from University of Balearic will guide you to the emergency exit. We have a total of 240 participants today from 20 European and 14 non-European countries. Non-European countries from Middle East, North Africa, other countries, non-EU from Europe, and North, Central, and South America, Australia, and New Zealand. We welcome all of you, and we wish you a fruitful discussion and to enjoy the conference and the networking.
Thank you. Thank you very much, Giuseppe. And now I give the floor to the director of the institute, the research institute, the very recent research institute in the Balearic Islands, Dr. Ippolito Medrano.
Good morning. Thank you for coming to Mallorca, land which is now under the pressure of the Xylella attack to very important crops in this area, like olives, vineyards, or almonds. And in my case, this institute, INAGEA, is an institute, a recent institute on agrobiology and water use efficiency, which belongs to the University of Balearic Islands, joined with the regional community of Balearic government, and it is very important, with the presence of the Instituto Nacional de Investigación y Tecnología Agraria, INIA, which is well known for you as a research institute for all Spain. The main objectives of this institute is to promote research of excellence in agro-food chain and environment, and also to devote particular emphasis to the research and development objectives of the Balearic government, and to facilitate the applicability of the new knowledge and contribute with the innovation in the Balearic agro-food industries. There are 5 departments inside the Institute: the agro-food production, soil and plant physiology, crop and animal protection and productivity, fisheries production, and water use efficiency by plants. In only 1 year, this institute, now with 26 doctors, all them belonging to the University of Balearic Islands or the regional government or INEA, has been able to produce a list of 14 research papers in the first percentile of the easy way for knowledge. Is now with interest in to promote the maximum research for agro-food industries, but also for water use efficiency, which means the main problem in relation with climatic change in the islands and Europe. We hope to extend our collaboration and to contribute also to the maximum quality research in this field of phytopathology with Xylella fastidiosa. I hope we'll be successful and will show some light for this difficult problem. Thank you very much.
Okay. Thank you, Dr. Medrano. And then I give the word now to Dr. Jordi Llabres, Vice Chancellor of Innovation at the University of the Balearic Islands.
Bon dia a tothom. Good morning, dear organizers, authorities, colleagues from the university and other research centers, participants from various universities across the world. It is for me a pleasure to participate in the opening session of this European conference on Xylella fastidiosa and to give you a very warm welcome to the University of the Balearic Islands on behalf of our rector and on my own behalf. Welcome to a land which, beyond the cliché of sun, sea, and sand tourism, offers you a historic, natural, and cultural heritage of great value, and produces also scientific knowledge which is called to play an essential role in our future. And in all humbleness, but with perseverance, our university is committed to this task. Not in vain, our university has become consolidated among the top 10 universities in Spain out of 83, as far as research quality is concerned. A remarkable achievement that some of the participants in this meeting have significantly contributed to. So, my gratitude to all of them. Thus, as an institution, we are strongly committed to high-quality research and to the transfer of the knowledge thus generated. Research implies production of knowledge, understanding why the things are the way they are. Research bears on, and it is the response to, one of the characteristics that makes us human, curiosity. Transfer of the knowledge generated through research allows us a better living. It engages, by its own nature, other stakeholders, administration, enterprises, and society. Therefore, it is a pleasure and an honor for us to host this European conference, whose main objective is clearly aligned with these two purposes: research and transfer of knowledge, that is, finding answers to a global problem, a serious threat to the agriculture and environment of Europe and the Mediterranean region. Thanks to our organizers, thanks to the speakers, and also to all the participants. Thank you for your work, and thank you for making this possible. Have a nice stay amongst us, and let your time and work here be very pleasant and productive in all sorts of ways. Thank you very much.
Thank you, Dr. Llabres. Thank you, all of you, for coming today to this opening. Thank you again to all of you for coming to this conference. I'm sure that the research that we will see during these days will shape, for sure, the management of the disease for the following years. So I do really encourage you first, again, to keep the schedule as possible, but then also to encourage you to very nice discussion and to trying to go beyond even our limits on what we are doing now on science. So thank you very much again for coming, and we close here the opening of the meeting. Thank you. So we start with the first session, and I really have the pleasure to introduce to you to Alexander Purcell. Sandy? From University of California in Berkeley. So, Sandy.
Thank you. It's wonderful to see so much interest from so many different places. One of the things about this get-together is it allows people who will never have to worry about Xylella in their country, but because of their scientific interests, they will make contributions. So I guess my job is to keep the train running on time today. We're short of time. We'll save all the questions for last. But we're going to, these are very appropriate to open this meeting and talk, first of all, an overall picture of some recent developments, and particularly in Xylella genomics. Then we have reports on the status in Italy and here in Spain, and finally Costa Rica, France, and then Costa Rica. So the first speaker will be Rodrigo Almeida, University of California, where I also am from. And he'll talk about new risks from an old threat.
Can you hear me? Perfect. All right, I see my choice here. Before getting started, I would like to thank the organizers, EFSA, for putting it together with Miguel and the university here. So thank you very much. I know it's a lot of work. Appreciate that. As I just started talking, but the clock already says 1:30, and I only have 25 minutes. Oh, Sandy's keeping track with the right time, so that's perfect. What I'll try to do today is give you some, in some ways, kind of my opinion or some data and some examples of things that I think are relevant to you today rather than sort of a standard research talk. So in some ways, this is sort of a synthesis of what's out there. Primarily focused on Xylella itself, because I think that's the most important part of the story here today. And I will say that I tend to speak fast, and my slides tend to be packed with lots of data. Please, each slide has a point, and it's not the data. The data are kind of just figures there for you. Please bear with that, or with me. So, getting started, first of all, I would like to thank people in my group. So this is a photograph of some of the folks in the lab. I'd just like to point out that we have received funding from a lot of different sources over the years, and that has influenced the work of what I'm going to talk today. We also work with many of you here in this room, all the way from Brazil, Costa Rica, different places in Europe. And I'm not going to thank everybody here. Just want you to know that or let everybody know that all this work has influenced what I'm going to cover here. So this is a brief outline. I hate to have to introduce Xylella in a Xylella conference, but I'll go very, very quickly through that as I'm the first speaker, so I feel kind of obliged in having to do it. I really want to focus on sort of the drivers of disease emergence. We're kind of starting to think a lot about that, but also about Xylella and kind of what does it mean as a name. And that's, of course, important for risk assessment and quarantine purposes. So obviously, we're talking about a system that has an insect vector, a host plant, And the bacterium, of course, the bacterium colonizes both the insect and the plant. It has to be able to colonize these two different hosts. Without that, you have no Xylella, essentially. So, you know, this is kind of something you've seen before. I also want to bring up the fact that you have lots of different layers of ecological complexity that you should keep in mind. So, of course, biotic and abiotic interactions will drive and determine how much Xylella you have in the environment. You also have to think about vector, pathogen, and host plant ecology. You have to think about the different outcomes of these interactions. Disease management, how you do that will also influence how much disease you will have. And what's important to keep in mind is that if you change one of these components just a little bit, you may go from a situation where you have lots of disease to no disease or very limited disease. And you can go from having no disease to having a lot of disease. So you have to pay attention that this complexity is actually complex, and you can't just look at these components in isolation. The second point I want to bring up is that Xylella is sort of an interesting pathogen in the sense that you have disease. Usually we're concerned about the agricultural system here, but you also have Xylella and vectors in more natural ecosystems. That's true in a lot of places in Europe. Is also true in a lot of places in the Americas. Okay, so in some systems it's more kind of simple, in agriculture primarily, but you also have vectors and pathogens often migrating from one to the other and back and forth. So Xylella, everybody knows, sort of xylem-limited bacterium colonizes a very large number of host plants. This list is going to keep increasing as long as you continue to do research. It's starting to pop up everywhere, and you only have xylem sap-feeding insects as vectors. So, although a lot of research has gone, primarily in the US and Brazil, and trying to understand how Xylella causes disease, we still don't really understand it. It's a line of research. There's a lot of interest right now. What we do know is that colonization of vessels leads to occlusion and water stress, and that ultimately leads to disease symptoms. Again, there's lots of different ideas here, how plants respond to Xylella, what induces plant responses, and so on, but that's not to be covered today. I also don't want to spend a whole lot of time on insect vectors, but we paid a lot of money for this particular movie here, so I'm showing it to you. If I show it every talk until I retire, it will kind of pay itself off. The point I want to make about insect vectors is that you have these essentially two groups of insects that are pretty well characterized in some ways. You have the sharpshooter leafhoppers, which are important in the Americas, not really particularly important in Europe, and you have spittle bugs here that are vectors. I want to make it stress this. You all have vectors. The question about vectors is not if you have them, it's not if they can transmit. They can all do it. That has been shown for 67 years now. That's not the question. The question is if you have one that's epidemiologically relevant. Okay, so you all have the vectors. That's not really something to be discussed. The question is, do you have one that actually matters and is important? And if you want to see, this is a leafhopper, of course. If you want to see the spittle bug, you can go to a poster by Emanuele Ranieri, a student from Italy, and he will show some of the movies for the spittle bugs as well. And Xylella transmission is something we spend a lot of time studying. And I would say that the only component here that I want to bring up that's important is that there is no vector species pathogen specificity. Essentially, all of these spittle bugs you have out here should be able to transmit all different types of Xylella. Okay? So the day that someone really proves that you don't have that specificity, let me know, because I'll be really excited. It's something that we talk about. But every time we run the experiments, you don't have specificity. So you all have the vectors, and they all should be able to transmit all Xylella. And that's, again, is not something to be much of a question. The question is if you have one that actually matters. So that's my incredibly brief introduction of Xylella. But again, to this audience, there should be nothing new here. What I said, I'll very quickly go over to why we're here. This is why we're here. I think that everybody has seen these photographs that Donato took at some point, and they really illustrate the problem. So Europe hasn't really thought very much about Xylella in Europe until October 2013. However, other people have. People working with Xylella before have actually thought a lot about Xylella in Europe and why it's not in Europe. It turned out that it has been in Europe for a long time. But here's an example. This is something that Sandy wrote 20 years ago. Many of you have read this one article. It's a review article, and it's essentially asking, why don't you have Xylella in Europe? And here are a few things that Sandy identified that are still very true, of course. First is that vectors are not a limiting factor. Again, you have vectors. Everybody has them. The fact that you cannot really see the disease or detect it doesn't mean that it's not here. So that's the standard sort of hallmark about Xylella. And the other one is that you should be concerned about introductions and avoid introductions, but if they happen, you should do everything you can to limit dispersal. So we're talking about containment. Again, this, should be no news here. You hear about this in every Xylella conversation. But it's something that, has been thought about for a long time. All right, so one of the things that we in our group have been thinking about a little bit are sort of how to think about drivers of disease emergence in the context primarily of Europe, but not only Europe. And disease emergence here, what I'm talking about is when you have an epidemic. It's not when a pathogen is introduced, or not when you detect it, when you actually have disease, okay? Or you have an increase in prevalence. So we came up with this sort of scheme here that's relatively simplistic, but it does cover a few components that we think are relevant. So you can have an endemic vector, introduced vector, you can have an introduced pathogen, endemic pathogen. You can also have pathogen evolution that occurs in different ways. You can have human-mediated dispersal, it can be of the vector or the pathogen, can be accidental. Usually, I tend to think that you're going to have here this accidental movement. You can have accidental movement by individuals. You can also have deliberate movement, which falls into a different category. We're not going to talk about that here. You can also have expanded geographical distribution, and that may be a consequence of land use change. So essentially, maybe management is different, or maybe you're watering your plants, or maybe you're just growing more of that particular crop in a new area. And of course, climate change is something that we don't have particular examples for Xylella, but it's something that we've been thinking a lot about in this context. So this seems very intuitive. In some ways, it should work, but if it's going to work, you have to actually be able to map different diseases on it. So here's an example of sort of this exercise. So here you have olive leaf scourge. You have an endemic vector. You do have what we think is an introduction, accidental introduction, human-mediated of an introduced pathogen. Of course, this pathogen appears to come from Central America, which does not have any olive. So you had to have some adaptation, something that if you want to, it has to have happened to go to olive. CVC in South America, this is how we think about it. We have lots of endemic vectors. There is some evidence of recombination between introduced and endemic pathogen. There is, of course, the plum genotype introduced into Brazil is an introduced one. So we tend to think that you have recombination between an introduced pathogen and an endemic one, and that led to disease emergence. And you have different examples here. The blueberry leaf scourge, you have endemic vectors. And in this case here, we think we have an endemic pathogen. And what happened is that in the area in the southeast of the US, blueberry production went from being kind of relatively small to being very large in the last 20 years or so. So we tend to think that this is sort of what's driving that particular disease. And introductions, of course, happen all the time. This is a meeting that Giuseppe actually brought up. The three people in this photograph here were attending that meeting. And what's interesting is that there's this World Expo. So here are people, the scientists participating at the meeting, and these are coffee plants that were certainly introduced at that meeting. So here we're walking to actually go into the room where the meeting is going to be, and you have this nice example of introductions. Ironic, but it's how it goes. So the bulk of what I want to actually tell you about is Xylella. What does it mean? It's a species, right? But what does it actually mean for you that is relevant? What kind of decisions can you make based on what you know? So first of all, I want to bring up the fact that Xylella, of course, evolves like any biological entity. Xylella is subject as a bacterium to DNA acquisition through different forms. You can deletions, reductions, optimizations, and whatnot. Xylella is absolutely no different than any other bacterium out there, okay? It's a bacterium. It happens to colonize insects. It happens to colonize plants. And most of the time, it does not. Xylella is hardwired to not cause disease, okay? Xylella does not want to cause disease. This is really well established. It causes disease, and the reason we think is sort of just because in so many ways monocultures kind of promote that. Something that I would be very happy to talk with anybody here, but the fact that the biology of it is hardwired to not cause disease, we have plenty of evidence that strongly suggests that. The one thing you hear among, we have conjugation in Xylella, you have the ways of acquiring genes, conjugation, you have transduction with phages, but the one that everyone talks a lot about is recombination. So recombinations, of course, we're talking about homologous recombination, so essentially switching alleles of the same gene. In the case of Xylella, it occurs, this is an analysis that Mathieu in our group just ran a few days before we, like last week or something. And essentially recombination happens more frequently than mutation. For Xylella, it's about 1 mutation in every generation of the bacterium. So recombination occurs incredibly frequently. This is some more recent work by Prem Kandel with Leonardo de la Fuente's lab, and he's tested all sorts of different things. And again, doesn't matter what you change, recombination is key for Xylella, and occurs very frequently in nature and in the lab. So the question is, does it matter? So here's a very busy slide, and it's an interesting story I want to cover. It's work by Euphresco in Brazil. And what you have in Brazil, you have citrus disease, and in this case here you have coffee and you have plum. The plum disease particularly is the interesting one I want to bring your attention to here. So the plum genotype that was introduced in Brazil is this ST26, it's a sequence time, so essentially it's a group of the genotypes. ST26 was introduced, but in southern Brazil and most of the plum plants in Brazil, you detect ST26, which is, let's say, a pure match or the right match to isolates from North America. It was introduced, but then you have 63, which is exactly the same, except that it has 1 allele that comes from endemic South American isolates. And then you have 67, which has 2 alleles that come from Pauca. And it's kind of interesting because you have a single introduction, and when you map these introductions into your phylogenetic tree, these 3 over here, here is in 3 different trees. It doesn't matter how you build the trees. These genotypes come up in different places. So here's one of them. Here's another one. I can't actually see the other one. Here's one. These 2 come together. Here's another tree. One comes out here, another, and the 2 are kind of separate here. So it's kind of interesting that you have a single introduction. So biologically, this has the same genomic background and everything. But if you run an MLST and you run different types of trees, they split, and you end up with the pathogen placed in different locations. What's kind of pretty cool here is that you also have a Pauca genotype that was found in plum, and this one, interestingly enough, has recombination with plum. Doesn't mean that recombination is driving this host switch here to plum from Pauca, but at least there is contact and presence. It has started. So everyone has seen something similar to this in terms of Xylella taxonomy. And at the subspecies level, and the question is, what does it really tell us? And it doesn't really tell us very much of applied value. Academically, it's important. It does help us think about Xylella in some ways. But once you go to the subspecies level, it doesn't really tell you anything, which plant it's going to go to or anything like that. So just want to tell you that at this level of resolution, it's not particularly helpful for risk assessment. So let's, you have different colors, different subspecies. You can go to Pauca. For example, this is based on current data. It may not be current anymore. In Palka, it's sort of interesting. You have a little cluster here of citrus that originates from coffee, different kind of genotypes. So it's kind of tight that in the sense that it's associated with citrus. Here you have different associations with coffee. So in this case, you have this group of genotypes that if you have something that falls within this clade here, most likely it will probably cause disease in citrus. If you have something in this clade, it will most likely cause disease in coffee. But that tells you about one host or a limited number of hosts, but doesn't really tell you everything, right? So you can get ST53, which I don't know how many plants it has been found to cause disease now, but 30? Okay, 30. And there will be more, right? So you can tell a little bit about the cluster here, but doesn't really help you very much in some ways. So if you try to get into more detail here in terms of resolution, STs are kind of helpful in some ways, but don't really tell the entire story. So let's go into ST53 because there is a lot of interest into that one. And here is genomic data for ST53. You have an outgroup here of other Palka. This is work by Sikar in the lab. She has a poster with her work. You can go check. So here you have some 40 isolates of ST53 from olive. It's, of course, a monophyletic group. We're starting to actually run out of ways to demonstrate that it's a single introduction. This is getting kind of pretty boring. But here, as an outgroup here, you have 3 Costa Rican isolates that were sequenced, and all of these are 53. Of course, of these here, 2 are associated with oleander, 1 with coffee, and these here with olive. So you can actually just go into the olive isolates and look if they have geographic structuring or anything. So here's the 40 isolates and where they come from and where they would fit in a tree. And long story short, so far there is no evidence of geographic structuring for this group. So you can actually really go inside an ST, and you still have lots of diversity, and the question is, what exactly does that mean? So if you want to summarize some of that in terms of the usefulness of the MLST scheme for risk assessment, this is data, but I don't have time for it. So essentially, if you have an ST, like you have ST1 in grapevines in Mallorca, ST6 in Alicante, we know that ST1 causes disease in grapes. We know that ST6 causes disease in almonds. You know it's going to go to those plants. That's probably going to happen every time you do it. However, you don't really know if it's going to go to other plants. You also have examples of phylogenetically related STs that may cause disease in the same plant species. So the citrus example I illustrated is one of those. But there are lots of other examples. You have closely related STs, and they cause disease in different plants. The same ST, however, can cause disease in different plants. So you have the ST16 example in South America that was in the previous slide. ST53 is probably the best example that the host range keeps increasing. So it's really hard to tell you, determine what a new host ST may go to. And of course, recombination may lead to STs that appear to be unrelated in a phylogenetic tree. Actually, they would be quite far away from each other, but they have a single origin, which is the plum example I brought up. And why is that? So here's an example from a preliminary analysis from 80 genomes of Xylella. And the tree, the details don't really matter. Essentially, it doesn't matter how you do your tree. You may have some differences in terms of subspecies and placement of different taxa, but you end up pretty much usually the same groups. What's really cool, though, is that here is a core genome. So this is 80 genomes, and here's the shared genomes for all shared genes in all genomes. It's hard to give a talk at midnight your time. So here's the core genomes, 600-something genes. You can put, maybe 800. So essentially, 1/3 of Xylella genes in any particular genome are shared among all genomes out there. And that, of course, explains a lot of the diversity that we know. So when we're talking about Xylella, we're talking about actually a bug that, stuff from Pauca in relation to multiplex or any of these, they actually share very little. And these are probably standard housekeeping genes that most bacteria have. Everything else is accessory. So if you count the number of genomes here, 80 at this end here, there are 700-something genes that are shared among all Xylella. The vast majority of genes are shared by single or very few isolates. So if you're thinking about risk assessment and predictions, the one thing that we kind of want to try to learn, and we still don't really have an answer, is this question of host jump predictions. So we try to address, we know intuitively that there is no relationship between host plant and Xylella in terms of coevolution, but you know, it's nice to have numbers. So here you have a tree for Xylella, and here you would have a host plant. So when you have coevolution, cospeciation, these two trees would line up. And of course, if you have, if this Xylella goes to this plant and this Xylella goes to this plant, so on and so forth, you have congruence among these, between the trees, and you could actually try to predict host jumps. It's something that has been done for fungi quite successfully. It's some really neat work on that. And of course, if you have no tree congruence, you're going to have all these different lines everywhere, and then you cannot predict which new host Xylella is going to go to. So these are the data, kind of pretty clearly what it is. And it's statistically significant. Essentially, there is no congruence between the trees. So the idea, at least today, that you can predict what host plant Xylella is going to go to, in addition to the ones that you have actually found it, is something that you cannot do. So I'm going to skip this, and I'm going to go finish here with a couple of messages. The first one, this is a nice slide that, of this very famous book that everyone in California has read at some point in their careers. Rodrigo Krugner took these photographs. It's really cool. And the message I have for you here is that Xylella has been in California for 120 years, and we still have a problem. We actually had the worst epidemic that we are kind of aware of, including Sandy, in the prime region of grape-growing California, in Napa and Sonoma Valley. We had very severe epidemic. And what I want to bring your attention to is that one program alone has provided, since 2000, close to $100 million in research dollars alone. This is only research dollars. And, all right, I'm going to go 1 minute ago, sorry.
Okay. How do we do this? There we go.
Thanks, Andy. Where was I?
You got -1.
All right, -1 minute. The message here is that for California alone, only grapevines, since 2000, $100 million invested, and really the only non-transgenic solution, which it doesn't really apply, the transgenic solutions don't really apply for you. None of them actually is out in the field there. The only kind of most exciting thing that goes beyond standard roguing of infected plants and pesticides is work that Andy Walker is doing with resistant varieties. He was going to be talking about that, and but you know, it's still kind of it takes a long time. Breeding takes a long time, right? So this is just a number there. Of course, it's not all Xylella research funded because it's a much bigger number than that. This is one program alone, and it doesn't include any efforts in quarantine and containment, which is much bigger than this number. So here are my unsolicited comments that I feel kind of obliged in sort of giving. So this is really not a plant pathology problem. This is not an entomology problem. It's not an ecology. It's not a detection problem. This is a complex problem. And unless you work together and you consider all these different components from these other disciplines, you're really not going to go very far. So it's a complex set of diseases, and management requires a holistic approach. And you will actually have to find your own solution for each one of the diseases in different regions that you have. This is a problem where one size does not fit all. You will be able to get examples of management from California, Brazil, different parts of the US, and so on. You're going to get ideas. You're going to have, you know, concepts or where to go, but it will certainly not, they will certainly not work for you right off the bat. You're going to have to learn about your own system, go out there and do the field work, and adapt and develop your own strategies. So I think this is sort of a key message and something that I want to make sure you guys sort of understand that even in California, Pierce's disease, different regions, you have different management strategies. Okay, thank you.
Thank you, Rodrigo. We're close to on time, but this is going to cut into our lunchtime, I think, if we go over. So I'm happy that you're trying to keep in the limits of the time limits we have. So now from Italy, Maria Saponari. If you've been to these previous conferences, you know that you always find some amazing data that's new and you haven't heard of. So Maria, I hope you have something new this time. Here's time.
Thank you and good morning to everyone. I want to thank the EFSA staff for leading the organization of this meeting and the University of Balearic and Miguel for the logistic organization. I will, in this presentation, I summarized some of the studies, the results of the studies that we have been done in the last 2-3 years related to the biology of Xylella fastidiosa. As Rodrigo remarked, it is not possible to predict the host, and so which would be the affected plants in the new outbreaks. So all the biology experiments are very important in order to determine which are the species that are under threat. When Xylella was reported in Italy in 2013, the first step was to determine the subspecies of the isolate and the strain associated to these new outbreaks. And we used the most common approach for Xylella, the MLST analysis, using performing it directly on the infected plants, because isolation at the beginning was a tough task to accomplish. Based on these preliminary results, we had evidence that the strain infected olives in Italy belonged to the subspecies Pauca, and the sequence type 53 was consistently associated to the infection. Later on, isolation was accomplished and successfully with the culturing of the bacterium, and so more extensive analysis have been performed, complemented with the NGS approach, which further confirmed that, as also Rodrigo mentioned, that we are dealing probably with a single introduction in the area, and that the strain causing the infection is the ST53. And based on this information, 2 years ago, we selected one of these strains, which now is our reference strain, and the genome has been determined, and this is the strain that we use for all our biology studies. The need to perform this biology and pathogenicity assessment in our situation was motivated mainly because olive at that time was a sort of a novel host for Xylella in general, but for Pauca in particular. The olive quick decline syndrome that we started to observe in the outbreak area appeared to be one of the most severe olive disease so far described for olive. And the sequence type, the strain, the genotype that was identified in our outbreak was not described before except a concomitant description in Costa Rica. So the biology of this strain was unknown, and thus it was very important to determine the biology of this strain. The approach we followed fell in these two categories: artificial inoculation and then vector-mediated transmission. The first type of experiment are performed under greenhouse condition, while all the vector-mediated transmission are possible because we have a demarcated area where we can work with the naturally occurring vector population. For the artificial inoculation, we choose the variety that is one of those most commonly found in the outbreak area and which showed severe symptoms in the field, so the cultivar Xylella di Nardò. And we inoculated different types of plants. Generally, we inoculate young shoots with at least 3 points of inoculation. The results of this mechanical inoculation show that in olives, for the colonization of the plant after the inoculation, the bacterium could be detected above the inoculation point at 5-7 centimeters only after 3 months, whereas in these plants we could detect in the apical part of the plants the bacterium only after 1 year. And at this time it was also possible to detect the bacterium in the roots. Symptoms on these plants started to appear soon after, so after more than 1 year, and were similar to those observed in the field, as shown in this picture that you may already see before. And so on the same tree, you can, especially on the plants that were bigger, we could observe the asymptomatic part with the shoots that start to wilting and desiccating, and then this typical color that the leaves have in the field, remaining attached to the affected branches. And these are the output, the results after 2 years on the plants of these varieties. As we observe in the field, there are some sprouting coming from the rootstock. Another approach that we followed was to graft infected scions from the field on healthy rootstock, and we also reproduced the symptoms of the disease, and not only the symptoms on the scion, but we detected the bacterium in the rootstock and in the roots, thus confirming that it was graft transmissible. We used these kind of studies also to determine the cultivar susceptibility, because following some preliminary observation in the field, we noticed some different response of the olive trees to the infections. And about this you will hear in the next session. And this is the results of the inoculation on plants of the cultivar Leccino, which is so far one of those that we identified as resistant. You see that there are desiccation, but the plants still continue to grow. And in this picture, after 2 years from the infection, the plants of Xylella di Nardò are completely dead, except the seedlings coming from the rootstock, while the symptoms on Leccino seems to be confined, and so the plant is still growing. Then we wanted to evaluate if infecting the plants using the vectors, we could also reproduce the symptoms. We used the Philenus spumarius collected in the free area, and we caged either on olives plant or on periwinkle, and then performed several transmission tests, including some on plants of the same varieties. We caged quite a large number of Philenus to be sure to have a high rate of infection. And after more than 1 year, only on one plant, we started now to observe some symptoms. So maybe the bacterial load at the beginning influenced then the development of the symptoms. So we are using this approach to evaluate the susceptibility of the olive germplasm, and currently there are more than 60 selections within the different projects that are under evaluation, and one of the tests is in the greenhouse, and the complementary tests are then done in the field. Because the results, because the inoculation takes a long time then to get the results, it is very important we are to use the controls in the inoculation, because we also experienced that some of the inoculation on this cultivar did not success. Thus, before to assess if some cultivars are resistant or not, it is important that in the test we include the susceptible one as a control. The experiment in the field to see how the disease progress and mainly how quickly the symptoms appear in the field, these are some data that we gathered in one of the experimental plot. The first year we caged the insect, while the second year this is a natural increase in the disease observed in the plot. As you can see, we confirmed that all these cultivars were susceptible to the infection, and the increase in the incidence is also supported by the fact that this is the diagram showing the infection in the Philanus spumarius occurring in the area. Regarding the symptoms on this plant, we started to observe some suspicious symptoms after 2-3 years, but at the beginning, of course, this small desiccation cannot be assigned to Xylella fastidiosa, but following during the time, the development, you can see that the entire branches died, and so it's more similar to what we expect. Here also another young plants that after 3 years show severe desiccation, and we are using these plants as, for example, as a source for the vectors. The major part of the experiments have been done on grapes because it was really relevant to understand the susceptibilities of the grapes. And we did as for olive field and greenhouse experiment, as you can see here, inoculation. We have completed the test for these 3 cultivars, and currently, with the support of the regional government, we have just started this summer artificial inoculation of more than 30 different varieties to confirm if they support the multiplication of our strain or not. And the results so far show that we could never detect the bacterium in the young plants we exposed to the vector in the field, and on the inoculated plants we could only detect the bacterium at the inoculation point. No movement was detected when we sampled the portion above the point of inoculation. Similar studies have been done for citrus. We tested some rootstock and some varieties. We have a field trials ongoing. We also caged some plants with the vector and artificial inoculation. In this case, I just remark that we got some positive detection using the vectors caged on the plants, but what we observed was that the bacterium did not move from the original portion where the insect fed, so it was not able to colonize the plants. And then a large program is also ongoing for stone fruit, because we know that cherry and almond, some few plants were found infected. but we extended the test to plum, peach, and apricot, and experiment included the tree approach, field exposed plant, caged potted plant, and artificial inoculation. For the stone fruit, we got only few almond trees, plants, and cherry infected upon the artificial inoculation, but after 2 years, no symptoms yet. For the caged plants, we only use this rootstock is GF677, and the reaction was like citrus. We could detect in the leaves where the insect fed, but no movement was possible to detect in these plants. And from the field experiment, very few cherry tested positive, but this year the same plants did not confirm this reaction, so this has to be investigated. And so far, plum, apricot, and peach we did not recover any infected plants. And just to conclude for cherry, because of our, as I mentioned before, that we had discrepant results, we are also following some plants in the field infected, all the plants, and we are seeing that every year, maybe because of the winter session, of the different winter season, we observed a delay in the symptoms, and this year, for example, we could barely see on the same plant some symptomatic plants. I conclude, and I want to thank EFSA and the regional government, because at the beginning they started, they promoted this first study, and based on this we acquired the skill and the collaboration with all the colleagues to conduct this work that then has brought us to this H2020 project, and all the team of the university, the CNR, and the Centro di Ricerca Basile Caramia for the work in the field and in the greenhouse, which is very important for this experiment. Thank you, and sorry for it.
Thank you. I'll remind you, we'll, excuse me, we'll postpone our question period or discussion until after all the speakers in this session. Our next speaker will be Dr. Blanca Landa from Cordoba, Spain, and she'll update us on some major points dealing with the Xylella in Spain.
I want to thank all the organizing institutions for giving me the opportunity to present the current situation in Spain. And I have to say that I will be the spokesman of the person that will present the results of many people and many institutions that are participating in these outbreaks. In Spain, the autonomous region is the one in charge of performing the official surveys and implementing the contingency plans. So when an outbreak is found, the different phytosanitary labs in each region have to provide the samples to the National Reference Laboratory from pathogenic bacteria that is located at IBIA in Valencia. And when the confirmation of diagnosis is shown, they send to our lab at CSIC to confirm the subspecies and to try to identify the different strains that we have. So the first outbreak in Spain appeared near the city of Manacor in October 2016, and was during official surveys, and it was shown that 3 cherry plants were positive for infection. Immediately in the infected zone, many samples were taken, and close to 2,000 samples of plants were eradicated. When these plants were analyzed, 4 polygala plants from the same garden center show positive. So when performing the NRST characterization, we show that 3 cherry plants and 3 polygala plants were located within the subspecies fastidiosa ST1. However, one of the plants from polygala was placed in the subspecies multiplex, but could not determine the ST because only 4 genes of the 7 could amplify. So immediately after, the buffer zone was monitored and many samples were taken. And from this point, they found many positive in the Mallorca island in different hosts and also in different islands. So in this slide, I want to show you the evolution through time of the different positive samples in Mallorca island. So in total, we have, if we add the 3 islands that we have positive, over 540 samples and 16 hosts. These are in red color, the positive through the 3 islands. You have a more detailed information in different posters from the government of the Balearic Islands. And the main positive are located in Mallorca, followed by Ibiza and Menorca. And concerning the subspecies identification, we had found subspecies fastidiosa ST1, one subspecies multiplex ST7, and one that is close to ST6 that show a nucleotide, one single nucleotide difference in CSIC gene, and is pending for numbering assignation and for giving a new allele number and ST number. We also found samples that were more closely related to ST73, but that showed 3 nucleotide difference in 3 genes. So they had provided a new ST number for this ST, that is number 80, and belongs to subspecies Pauca. So this is the situation of the different subspecies and the different islands. We have, as I told you, more samples, positive sample in Mallorca, with 3 ST and 2 subspecies, fastidiosa multiplex. In Menorca, we have found only subspecies multiplex ST6-like, and in Ibiza, subspecies Pauca ST80. Concerning the positive sample, most of the Mallorca positive are from wild olives and from almond. In Ibiza, the highest number of positive are in olive and wild olives. And in Menorca, the highest number belongs also to wild olives. The assignation and the different subspecies on host is the one that shown here for the different crops. We had found subspecies fastidiosa in grapes, almond, and cherry. We had found subspecies multiplex and Pauca in olive and wild olive. We have found subspecies multiplex in Ficus carica, and we haven't been able to assign any subspecies yet to plum or walnut. Last week we found also in Juglans region. Concerning ornamentals and natural vegetation plants, we have Polygala myrtifolia. We have found all the subspecies and all the STs. In Frasinus, we have subspecies multiplex. In Lavandula and Acacia, we have multiplex and Pauca. In Rosmarinus, multiplex. In Cistus montpelliensis, subspecies fastidiosa. And in Nerium, oleander, and Rhamnus alaternus, we haven't been able to identify the subspecies yet. Here I want to show you some symptoms of wild olives in Ibiza. Mainly they are found very affected trees in abandoned orchards or in particular gardens. You have here also typical symptoms in wild olives. You can find in the landscape of the island that there are many wild olives desiccating and dying all over the landscape. And originally they thought it was due to the droughtness period, but now they have seen that they are positive for Xylella infection. Here we have typical symptoms of almond trees showing the golden death at the end of July when we were visiting the island, and the typical leaf score symptoms. And you have also the patchy incidence of trees, so you can have very affected trees and trees close by from the same variety that are negative, non-infected. These are the typical symptoms in grapes, in red varieties and white varieties. And just to give you an idea, about 60% of the sample have been already eradicated. Many of them were in the garden center, and some of them have been also, as precaution measure, have been also eradicated. Concerning the outbreak in Alicante, the first one was detected at the end of June. Early after, 1 kilometer far away from the first one, another field plot from almond trees were detected a positive sample, so the marketed area was higher than 30,000 hectares. Here you have the location of Guadalest and Benimantel. So the landscape is terrace and mountains and a valley in the middle. This is the aerial picture of the first plot that was completely eradicated in Guadalest. We could assign, in this case, the species to multiplex and the ST6. After a few months, 26 more plots of almond were found infected, and the demarcated area at the end of August was higher than 100,000 hectares. We have identified a subspecies as multiplex, but we have been able to finish the ST assignation yet. And just the last record we have is at the end of October, in which 58 almond plots were found positive, so the demarcated area is increasing. So in total, again, the subspecies multiplex, but the ST is not finished yet. So we had found more than 100 positive samples, but only in almond. Even the sampling has been focused on olive trees and other potential hosts, but so far only almond had been found positive. 86 plots, the eradication has already been accomplished in 19 plots and more than 500, close to 600 trees. These are the symptoms, very similar to the ones that we can find in Mallorca on the almonds. So currently, this is the genetic diversity we had found in Spain. We have 5 ST, or sequence type, and 3 subspecies. So we have the bad luck of having the highest diversity in Europe at the moment. What we are doing now is, because ST6-like is not exactly the same that the one that we had found in Alicante, so we are checking different genes of, so far they show identical sequence, except the CSIC gene. Also, in collaboration with Maria Agnès from IRA, we have seen that the samples of the different subspecies, only the fastidiosa subspecies, harbor this plasmid that is involved in conjugation and DNA transfer among bacteria, but is not present in the other subspecies samples. Also, we had found that the ST80 from Pauca in Ibiza has the unique open reading frame sequence that was shown as specific for mulberry strains, but is also present in other Pauca strains. So we found a big difficulty to identify the different subspecies because we were working directly with DNA extracted from plant extracts. So about 45% of the samples were finished up to the sequence type level. But we found some problems of inhibition, or probably the CT values were very high, so the bacteria concentration was low. Also a problem we faced was that the most of the plant of the samples of the first outbreak were processed at the end of winter, early spring. So probably also the bacteria titer was lower because samples taken in summer we have more success in the identification. And another problem is that many of the plants were eradicated, so we have no more samples to process. We are now revising the EPO protocol, and probably we believe that more than 70% of all the samples that we have analyzed will be at least positioned at the subspecies level. The strain isolation was initially low, but now is becoming much better, and probably this will allow us to sequence the genomes and having more information. So just to conclude, we have found new host plant of Xylella in Europe after these new outbreaks. This is the case for Ficus carica, Frasinus angustifolia, Juglans regia, Prunus domestica, and Vitis vinifera. Also, an important fact is that although ST6 is widespread in France, in Corsica, they have not detected in almonds. Only, I think there is only one case, whereas it's the only host infected in Alicante so far. So probably we are just looking to the ST type, but maybe the strains are different. We have also found new subspecies and sequence types for Europe. This is the case of fastidiosa ST1, subspecies multiplex ST6-like, that I have found the same sequence SNP in a case in olive in California, so I don't know if it's similar or not. Subspecies Pauca ST8 is new for science. So our idea is that several and independent introduction has occurred in Europe and Spain in the past, and from different origins, probably from the USA and Central America. And the sequencing of the genome of representative isolates can provide insight into their origin and biology. And finally, I want to thank all the people from the government from Islas Baleares and from Valencia. I cannot name all of them, but this is the real people that is doing the sampling, the isolations, and it's a really hard and intensive work. I want also to thank my colleague, Esther Marco, that is the head of the National Reference Lab of Bacteriology, in which I have a close collaboration, and we are learning one from each other, and my two colleagues from CSIC, and of course of the project that we are participating. Thank you for your attention.
Thanks very much. Now we'll get a briefing on the update with Xylella from France by Françoise Poliakoff from Angers, France.
Thanks to the organizers to give me the opportunity to present the Xylella fastidiosa situation in France, mainly in the region of Corsica and Provence-Alpes-Côte d'Azur. First of all, the story of Xylella in France began in 2012 when we discovered on intercepted coffee Xylella fastidiosa that we could confirm by isolation after 1 month to get few colonies and immunofluorescence. So we needed at this stage, facing the introduction or the risk of Xylella, to evaluate different methods available in the publication for validation of a good, reliable method able to early detect the diseases and to provide for monitoring and survey and control. So we go through the validation scheme with following the EPO protocol that is proposed by EPO for validation. First of all, it's the standard EPO PM798, and we have at ANSES a guide for validation too. We went through inter-laboratory evaluation to evaluate the performance criteria like analytical specificity, analytical sensitivity, diagnostic specificity, and diagnostic sensitivity, and repeatability. And then we went through inter-laboratory evaluation with a partner for EU, all European laboratories, and to validate the methods and then be able to transfer to laboratories and train laboratories to apply this method. So for this evaluation, we generally use sample that we spiked with dilution, different dilution of suspension of Xylella fastidiosa. We work with all the strains, but as Rodrigo said, there was no differences between the strain. And we then perform with different matrices the PCR after DNA extraction on different replication and on different days. So we could validate it this way, the method of Harper, to propose for the survey and official control. With DNA extraction automatized with a quick pick plant DNA quick from BioNobile and Kingfisher device. So I show you very quickly the performance criteria of this method we choose to develop in France. You can see that we determine inclusivity and exclusivity. That means specificity on quite a lot of strain. Sorry. Here, 45 strain, and on non-target, on 29 non-target. And we got quite good sensibility, analytical sensitivity, 10 to 2 bacteria per milliliter on orange tree, on grape 10 to 3, on olive tree with a quick pick and the multiplex, maybe the mixed we use, we got less good sensitivity. So we are working more on this fact now because there are quite a lot of inhibitors with olive tree in this case. So this method helped us to detect for the first time in 2015 the case in Corsica on Polygala myrtifolia. Since then, 15 samples have been prepared and analyzed, and we got around 900 positives, so a rate of positive sample of 6%. And so we observed that more than 50% of the area included is included in a demarcated area. Outbreaks located more in urban places, private and public gardens, and natural areas, maquis forest, difficult to access. As you see, it's quite a lot of mountains and hills. So the strategy of eradication is not really adapted for Corsica, and the containment strategy will be adopted by the end of 2027. So the main host plants which are positive in Corsica are Polygala myrtifolia, that represent 54% of the positive cases. Then we have the Cistus montpelliensis, which represents 6%. We have Helichrysum italicum, which represents 10%, and Calicotum, which represents 11%. So the repartition of positive hosts is described here. And the sampling was made on 327 species. In Provence-Alpes-Côte d'Azur, we discovered also Xylella in 2015. We, since then, sampled about 5,000 samples, and we got a rate of positive of 2% in this region. The most plant host find positive is always Polygala with 60%, and Lavandula 4%, and Spartium gentium 20%. In this place, outbreaks are located more in urban and peri-urban areas, private and public gardens, so the strategy of eradication is still ongoing and still successful. To see here the top 10 of the host plant in France is more, as I said, Polygala, with a more sample host, 2,700 sample, and with a rate of positive of 19 in all the Polygala sample. Calicotome is a very high contaminated also host with 33% of positive. And then you have, as I said, Spartium gentium, Helichrysum, and Cistus montpelliensis for the 5 first. And then for the top 10, you have still Lavandula, Cistus also, and Genista corsica, but with a rate of positive lower. So we got at this moment 38 host plants for Xylella fastidiosa. So at national level, to conclude, there was 24,000 samples, a lot of species sample, but no detection of Xylella in cultivated zone at the moment, nursery, orchard. More than 80% of contaminated sample belong to endemic plant species from Mediterranean flora present in Marquis Corsica, and since 2 years, boundaries of demarcated areas do not evolve so much. For identification of strain of Xylella in France, in collaboration with Marie-Agnès Jacques team from INRA, we developed the identification scheme by MLSA, MLST, based on the PCR, Yuan and Tal, to amplify 7 housekeeping genes. And we could assign most of the sample to the subspecies multiplex strain. And going through MLST analysis to identify the sequence type, we found always are finding sequence type 7 and 6, which are have high identity with the USA strain multiplex. So is to show you how is the repartition of the strain in Corsica and Provence-Alpes-Côte d'Azur. At the moment, you can see on Corsica mainly sequence type 6 in the north and sequence type 7 in the south, but a place, a zone where there is a mix of the two sequence type, multiplex, and in some sample we found both sequence types 6 and 7. So we found the coefficient. So we can imagine there was at least 2 introduction of strain in Corsica, but maybe more, and when, that is the question. In south of France, we also found 2 subspecies, but we found one location with subspecies Pauca sequence type 43. Just on 3 plants which were eradicated, and we didn't find any more this sequence type. So to show you here how when we apply MLST on multiple to identify the subspecies and mainly on multiplex strain, depending on the host plant, we are more or less successful in assignation or assignment of subspecies. For example, here in Calicotum, sorry, it's
1 minute left.
How I can go back to the, sorry. Depending on the host plant, we could no, it's no. Okay, I will manage. Sorry, I am always doing this. It's my specialty. Sorry. So here. Good button. Here we put the successful MLST on 88 sample in 2016-2017. We were able to have successful MLST on 29, so on 33% on Calicotum. Here you have the CQ value. So on Calicotum, here is the failure, the percentage of failure, it's quite high, 51%. On Polygala, the failure is very rare, so we got a very successful result in MLST, 85%, and so on. So you can see here that Helichrysum, Spartium is quite good, but when we go to Lavandula and Cistus, the results are very poor. So that's why I invite you to see our poster where I present how we manage with that to improve the quality of identification. So the perspective for our work now is evaluate and validate other methods to improve Xylella detection on complex matrices, for example, digital PCR, identification of Xylella by MLST, in collaboration with the EPO group and POnTE partners, improve detection in vectors, and we propose at this period test performance study that is ongoing to evaluate different methods on vectors. And we will have to evaluate detection keys to use in the field because there is a very high demand on this. So I want to acknowledge all my staff and colleague Pauline de Gerfagnan, who has a poster too here, colleagues of Ministry of Agriculture, Juliette Oricost and Sausen Joudard, who is here also, and partners of INRA, the team of Marianne Jacques. Thank you very much.
So our final speaker in this session will be from Costa Rica, Carlos Chacon, and that's very interesting to see someone talking about the El Dorado of Xylella. So that's, I'm sure you're going to tell us about that, Carlos.
I hope so. Well, I want to start by thanking the organizers for giving me this opportunity. So I'm going to try to give a historical and contextualize Xylella in another region, a tropical country. So I'm going to start with something that is important, and is that Xylella fastidiosa is not new in Costa Rica or Central America. This is the first report of Xylella in Central America, and as you see, the bacteria didn't have the name Xylella at this point, but it was clearly a Xylella. This was done from people from the south part of United States that wanted to grow grapes in Costa Rica, and the plants developed Pierce disease symptoms. So they took them back to California, and this is the first photo, or the oldest photo I know, from Central America of Xylella fastidiosa, these photos. This is important because I'm going to try to contextualize that situation in Costa Rica, but this is recurrent. You get probably a lot of vectors, probably a lot of plants that have co-evolved with Xylella that do not manifest symptoms, and most of the problems are when you introduce sensible host plants to the country. And this is one of the first examples of this. For people that are not very familiar, this is the Central American region. It's a tropical region. It's conformed of 7 countries. The Central American region is a bridge, a cultural, geological, and most important, biological bridge. So we tend to mix things. Things from the north, things from the south, they tend to get mixed in Central America, and this is not is a case also for Xylella. In terms of biodiversity, all of these countries have the same degree and show a lot of similarities, historic ones and productive ones, and biodiversity. In the case of Costa Rica, Costa Rica is really a very rocky country. We have most of the countries between 900 and 1,000 and 1,800 level above the sea. We have 2 seasons, the rainy season and the rainier season. No, a dry season, okay? It rains a lot in Costa Rica, and the temperatures throughout the country are really stable. This may be one of the conditions that helps that we don't get a lot of symptomatic plants, because temperature is very stable throughout the year. What happened then? Some of coffee-producing people around 1990s started to see that the coffee plants had something that we call crespera, that in the traduction is leaf curling, and deformation of leaves. This is a mild disease in Costa Rica, but it was one of the first attempts to start looking for a cause for this. And the first report of this is in 2001, where it was reported that this disease was caused by Xylella fastidiosa. As in the right, there is a map of the country, of the regions where coffee is produced in Costa Rica. Costa Rica is a major producing country, coffee producing country. And as you start to survey this, you see that Xylella was found in all 8 coffee producing zones. The only thing is that Xylella was, if you look for Xylella, you find it in old coffee plantations or plantations that are not aggressively productive. In these productive productions, plants don't last, they last from 3 to 5 years before they're replaced. So this, as Rodrigo was mentioning, you have to tailor how to manage Xylella. This is how coffee growers have been dealing with Xylella, because the plants are very short age, and this doesn't help or doesn't affect the production of coffee in Costa Rica. But one thing leads to another, and so in Costa Rica, coffee plantations are used with shades, with trees. So you start looking for Xylella in all these shades, trees, and nearby plants, and you find it everywhere. So Probably the scenario is that Xylella is really endemic and has co-evolved with a lot of hosts, American hosts, and these are what predominates. The problem is that coffee is an introduced plant from the 1800s. So coffee is, again, like this grapevine that I was talking about. It's an introduced crop. Where we really see symptoms and plants really die is with oleander trees and Catharanthus. This is more recently. These trees that are used in gardens, they really dry out and they have to cut them. But apart from that, we only see mild symptoms and or no symptoms at all. In fact, when I was starting with Xylella, I almost went crazy because I tried to look for symptoms everywhere, and everything for me had symptoms, and it was something a little bit frustrating. But I understand that Xylella was everywhere, and that this is in Costa Rica. The scenario is that Costa Rica, that we have Xylella strains, we have vectors, and the problem is when you introduce plants. Some work has been done in vectors in the past. This is a list of some subfamilies of vectors, leafhoppers, and in this article there is a list of the specific vectors that have been listed and found related to the leafhopper family. Some dynamics have been done, but no specific vector studies have been done. And as Rodrigo mentioned, I think in this scenario, this is not truly important because we have a lot of these insects biting and keeping a pressure on the plants. In terms of genetic diversity, Costa Rica relates to Xylella fastidiosa, subspecies fastidiosa, and here are some of the STs that have been reported. One case that is of interest is the finding of ST53 in oleander in Costa Rica, and this ST53 relates or is to the Codiro isolates. They're very close together. With Rodrigo and the X-Factor effort, we're trying to make a template for the Costa Rican samples so we can have more information about this. So you see we have a lot of vectors, a lot of asymptomatic plants, a lot of native positive plants, and the problem or where we see symptoms is when you introduce this, some sensible host to the country. In our effort, we're trying to phenotype some of these strains that we have, because at the end, the bacteria will infect the plants, and we'll see that genetically it may be associated to some of these S types. But we want to see if we can make a description or phenotype these strains. One of the things we're doing is the problem with phenotyping is that it can be lab dependent. So we're trying to adapt some techniques that are used to homogenize this, and this is just one example. We're using the Bitec Biomeriux profiling that is normally used for another type of bacteria, but we're homogenizing the protocols to use it to have a biochemical profile of our strains. What you see here is we tested all our strains and have how many are positive or not for these tests. So we see that we can get a core of tests that are either 100% or 0%, and that are important to us. And when we compare with other bacteria, we see that these cluster together. So this may help to homogenize this interpretation and maybe relate with some genomic data. We are also fatty acid profiling our strains, and eventually it would be interesting if we could, these are only our Costa Rican strains, if we could add some strains from other parts to see if this technique allows discrimination between this profiling from strains from other parts of the world. We are doing biofilm assays, in vitro biofilm assays, and what we see is that the plants that are more symptomatic are the ones that produce less biofilm in our in vitro studies. And this is an interesting remark for us that we'll have to try to find an explanation. Another thing that we're doing is Costa Rica, coffee is important in Costa Rica, and I have a colleague in my university that it works with coffee rust. Coffee rust is really the pathogen that kills coffee production in Costa Rica in a short time. So he developed a model to study coffee rust and had good results. What we are trying to do is use this model for the Xylella question. We are using this variant of coffee is the one that the Institute of Coffee distributes, and when we go to the coffee areas, we find Xylella in this species. This one is the one that they're working with to implement for resistance in coffee for coffee rust. So we want to see if it protects against coffee rust, would it protect against Xylella? And this would be a very good, if it works, it would be quite good because these people from the ICAFE, from the Institute, are the ones that are managing and taking decisions. So if they change the host, it would be nice to know that we won't have Xylella in this coffee also. But as I told you, that coffee, intensive production of coffee doesn't really have this problem because the plants don't last that long. So we have a lot of Xylella circulating, different kinds of Xylella. We have a lot of vectors and a lot of diversity and a lot in numbers. You just take a walk and you see a lot of these kind of vectors. So you have a lot of pressure on the plants. Where is the problem? When you insert one of these susceptible hosts. And coffee, we have a history of coffee, but at the end, it is an introduction. So internal impact of Xylella in Costa Rica, I would say, we don't really see it. Where is the real problem? The real problem came when the European epidemic broke out, because one of the economy in Costa Rica, one of the parts of the economy is to export ornamental plants. So when the measures were taken, the exportation was closed, but I'm going to say closed. It's not really closed, but you have to have some regulations. And this is that you have to have the plants from 200 down meters free of Xylella in a country where you have so many biodiversity, and a lot of this is practically impossible to accomplish that. So the impact has been economical and social, because we have 4 million, almost 5 million people, and a lot of people lost their jobs because this happened. I know this is just to make a comparison of how can a pathogen can impact a country. There were some efforts to enlist this plant, but there has to be more scientific knowledge, and this is why we're collaborating from the stakeholder. The phytosanitary authorities are asking us if we can work in this question. Finally, I want to acknowledge these are some people in the lab that are working with these two projects. Some people in the past that have made a major contributions and are still working on Xylella. These are the two centers that conform this, the people that work on Xylella. Some other colleagues from the School of Biology, the phytosanitary authorities that we are working on. It's important to have them with you. and people in this ornamental exportation category. And that would be. I don't have.
Thanks very much. Could I ask the speakers to all come up here and sit behind your name for the discussion period, which we're limited to till 11 o'clock, so we have 20 minutes, unless the Jefe tells me that we can go a little longer. Okay? Yeah? Okay. We don't want the food to get cold. So if you have a question, please raise your hand. I know there's going to be one right here on the second row. And I think you'll have a microphone.
Okay, thanks. I have a couple of questions for Carlos Chacon. One, did you see some differences between oleander varieties in the expression of symptoms? And the second question is, am I correct if I understand that you noted more biofilm when the strain induced more disease? Is this the correct interpretation?
I'm going to start with the second question. Yes, interestingly, we did these biofilm assays to try to see. The question was, if you produce more biofilm, probably it would be more pathogenic. But the real deal is that as these plants that were really symptomatic, that really died, were the ones that produced less biofilms in our experiment. And we did it a lot, quite a few times, and this is what we get. The other question is, yes, in Costa Rica, the oleander is used in gardens. And we see that the susceptible ones are the ones that are white. The other ones, there are a lot of like pink tones. No, and when we go to look for Xylella, the ones that are we see symptoms and are positive are the white ones.
Okay, next question.
Well, you show a tree with the ST53, and I couldn't see much of the details because my eyes are not very good, but you see some grouping inside the ST53. Was that tree made with whole genome, and where do you see the difference, or how many groupings do you see?
So that's the tree with the I need coffee. That's the ST53 only tree, right? Yeah, so that's whole genome sequence data. The outgroup is 3 or 4 isolates of Pauca from South America. Then you have the 3 other ones from Costa Rica that were previously sequenced, and some 40 or so of isolates that Maria sent us. Is that her question?
Well, I'm talking about seems to that you see grouping inside ST53.
Yeah.
So is that variability just in Italy or compared to Costa Rica? How it compares?
Oh, how it compares to Costa Rica. So as a population, there are a couple. So if you map it against the reference de Dono, all Costa Rican isolates have about 150 SNPs or something in relation to all Italian isolates have about 150 SNPs in relation to the Dono, and the Costa Rican ones have an extra 50 or so SNPs. This is based, of course, on the things that align. I think Annalise is going to have a slide with the table, and there is a poster with all the numbers as well. So the number of SNPs is relatively small within Italy and in relation to the Costa Rican outgroups, which is a case for a single introduction. But they're there, yeah. Does that answer it? Yeah.
Yeah.
Sorry, Rodrigo, another question.
I want my coffee.
You showed us, and you just mentioned the diversity of ST53 in Italy. and you mentioned that there's no geographical clustering.
Mm-hmm.
So what is your interpretation in terms of epidemiological spread then?
Yeah, so when Anne brought the Montel results that said that there was no structuring, I asked her that question. And and it seems to fit a little bit of some of the model that the Bari group has been proposing, that you do have some short distance dispersal and you have some long distance. I mean, of course, within the context of this relatively small area. So you have the short distance dispersal and you have some long distance of these isolates, and once you do only 40 samples, you end up without any spatial structuring. You will see a talk in the next session or something about very similar work with a lot more isolates from California, and in that case, you do have some of it. So,
okay, question in the back.
This is a question for Blanca Landa. Is there any hypothesis on the origin of the outbreak in Alicante, in Spain, and also to the fact that in the Balearic Islands there are the 3 subspecies?
My hypothesis is that it was introduced by plant material. We have in Balearic Island ST1 that is commonly found in almonds in California. I know that many years ago, 90s, there was a lot of introduction of almond varieties for new plantations. So probably this is the best explanation for the ST found in almonds. Concerning Pauca in Ibiza, I have no idea because this is a new genotype and is closely related to the isolate from Central America, but I don't know. And in Alicante, the fact that it's only found in almond trees probably can be explained by the same fact that it was introduced with plant material. So, I don't know. It's difficult to guess at this particular moment.
Yes, this question is for Maria Saponari. You told us you need about 2 or 3 years to see symptoms in young plants, olive trees. Maybe can we extrapolate for all trees or not? I mean, do you have any interpretation on the incubation time on olive trees?
No, not exactly, because sorry, there is also the problem of the on this huge canopy of the ancient trees. We have, if in the area there is no vector control, we have a continued reinfection of the same plants. So we don't have exactly an exact idea about the time lapse that occur between the infection of an ancient tree and the colonization of the entire trees, and then the appearance of the symptoms. In these young trees, in this plantation, we are not using any vector control, and so the symptoms appear more than 2 years, and but any comparison with the situation in the ancient tree is still difficult.
Thank you.
Yes, thank you. It's a question for Mr. Chacon from Costa Rica. So, in Costa Rica, you have a huge diversity of Xylella present. On the other hand, the environment is in such a way that expression of the disease is not promoted because temperature and rain and so on. But then you speak about two plants, oleander, the white oleander and Catharanthus, which show disease symptoms, although they are in the same environmental conditions. So maybe Costa Rica could be a good laboratory to test the real plant genotypes that are more susceptible to the disease, unless when the conditions are optimal for survival of the plant, but disease expression in some plant genotypes. Is that an idea?
It really, with the case of Nerium, this was seen years ago. But for this project, we are using the vinca plants as our control, because when this is a common plant in gardens in urban San Jose, that is the capital, and we see it a lot. So we're using this plant as an indicator. We go there, and they show a lot of symptoms. So, and we have isolated 2 strains from vinca, and we're using immunofluorescence for our survey test, because we think if we use a more sensitive method like PCR or ELISA, they will be more positive. But our goal is not that. Our goal is to isolate the strain. So I didn't explain in detail, but we get a lot of these vinca positive, immunofluorescence positive strains, and we see a lot more than from 10 to 20 per field in microscopy. So this is what we're using it for as an indicator, and it's really a susceptible host.
I have a question for Maria Saponari, and it is about artificial transmission with vectors. Did you observe changes in time, temporal changes along the year in your success when doing these experiments?
Yes, as I mentioned, we are carrying out this experiment in the demarcated area and mainly under field condition. And the problem is that we need to accumulate more years of experiments to get some conclusion. But one thing that we observed since the beginning was that in the hot season, in August in particular, the transmission was very low. We don't know if it's because in the plants the bacterium is not very active, or if it's a combination of the interaction from the plant and the bacterium. For the rest of the period, it's quite similar, the rate of infection that we get.
Okay. Question to the far back on the left, right there. Yeah.
If it's necessary to detect Xylella no more 3 months, as is possible to conserve a prebasic material in open field and not in a screen house? In general, the question is not for, in general.
Yeah, you mean that because in the symptomless, because the infections take a while.
It's possible to detect 3 months later in the lab. It's correct. I understand. So.
Yeah, and I think there are all the actions for the nursery that are located in the infected area in order to avoid that the material is produced under field condition with high risk of infection.
Because the European legislation permits to have a pre-basic material in open field and not in a screen house. So I am nursery problem. I am not sure that the material in open field is free or not of Xylella.
Yes, this is a potential risk, even because we don't know when the insect has, has visited the plants, and so when could be detectable.
What do you suggest?
Sorry.
So it's it's not better to conserve olive trees in a greenhouse. It's better than open field.
Yeah, I believe that if any certification process has to be done in some area, it is very important to avoid any risk that some material can be moved, and so to use all the precaution, like use screen houses to grow the mother plants and so on.
Thank you.
Okay. A short question to Françoise Poliakoff. Did the Xylella positive spartium plants show some symptoms? The ones that you reported as positive in Corsica and Paca region?
Yes, in some cases, yes, there are some symptoms.
And were these symptoms typical of Xylella? Some leaf scorch or something like that?
Yes, but typical of Xylella, we cannot say typical of Xylella. Of course. It's dry, yes, leaf scorch and dryness of some branches.
Okay. While we're waiting for the next question, I want to ask our three speakers from Europe, without showing anyone else, to write down your estimate of how many years it's been in your country. I realize it's going to be a wild guess. I want to see what the difference. Don't talk to each other. Next question.
Yes, I have a question for Blanca. You described the situation of the Balearic Islands, I'm here, sorry, and where you had more sequence types found in the Balearic Islands. Are there cases where these mixed infections or co-infections are found in several plants? So more than one sequence type, more than one genotype in one plant, in one individual plant?
We have only one case in a polygala plant. But so far we have not found mixed infection. Yeah.
Okay. And I would like to extend this for the colleagues from US and Costa Rica. Is this phenomenon of mixed infections or co-infections very common from your experience in the Americas?
I wouldn't say common. It does happen. The part of the problem is you certainly do have lots of infections. So if you use population genetic approaches, which together with Euphresco in Brazil we have, you can show that plants probably have multiple infections. It's probably what's happening in Italy as well. But if you do MLST, it's going to tell you that you have this one infection, right? So it kind of depends on your level of resolution or what your question is as well. So, but yeah, I think J.C. Chan from USDA, for example, he showed that you had multiple infections in almonds in California. Some stuff that I did as a long time ago kind of had some of that too. So, yeah, it's not very common, but you can have it. At least it's not commonly detected.
We have time for one more question.
Thank you. Question for Rodrigo. I just want to know, after 100 years in California and $100 million research dollars invested, can you show some positive examples whether this battle can be won, or we will continue to showing only the problems but not that much of the solutions?
So that's a somewhat loaded question. California is, if you want to focus on grapevines alone, for example, California is a major producer of grapes for wine and table grapes. It's a huge, I think it's the second commodity in the state. Andy, it's billions. And you have Xylella everywhere in California. You have Xylella everywhere in the United States. So as an example, Sandy had a little bush in front of his house, and he brought it in, we sequenced it, and it's a new, very divergent type of Xylella. So yeah, in Europe, you're gonna learn how to live with it, and that's where you are. You know, that's just very realistic. You're not gonna, plants will not disappear, and agriculture will not disappear. You're just gonna have to learn how to live with it, and that will be local solutions. As to the $100 million, in the last 20 years or so, there was a major push in that particular program for transgenic plants. And there are transgenic plants that appear to work under field conditions. Really, the only game in town for plant breeding is Andy Walker, and he's here, and he's going to tell you about his work. And I think you will find fairly remarkable what he was able to accomplish in terms of grapevine breeding for Xylella tolerance resistance. So I'll let him, summarize that. But for that particular type of work, there's been a lot of success. We also learned a lot about how Xylella causes disease, how it spreads. It's been, I would say we learned a lot, yet in the last few years we had the worst epidemic that at least Sandy can remember in Napa and Sonoma and other regions in the States. So we've learned a lot. We would probably be in a much worse situation if we hadn't done all the research and we didn't have all the experience, but it's still a problem, and will most probably continue to be a problem. And so welcome.
Okay, I want to ask our three Europeans here to write your number on a piece of paper. Don't show anybody else. How many years do you think Xylella has been in Europe?
Do it.
Yeah, I want you to simultaneously hold up your secret number. Secret. Okay. Okay. All right. I realize this is a wild guess, but I just want to get some feel for it, because these people do have some experience with seeing the data and having some ideas about it, perhaps. Okay. Can you all show me your number? 15 to 20 years, 25 years. 20 years. I would only say more than 20 years. So one thing to keep in mind is that what's expanding diseases. So perhaps what the disease is not spreading, but our recognition of where it occurs is what's spreading. So think about that. Well, thanks very much for all our speakers, and I guess we have lunch, and you have some announcement.
Just a very short announcement, please. All the speakers, there is a desktop there to drop your presentations if you haven't sent by the tool by EFSA. And also for the next session that is chaired by Blanca, I would ask the speakers to be here 5 minutes in advance, just to brief them about the time and so on. Okay?